Hello, this is Abraham Seka speaking. Today we are just going to go through the animal cells and try to explain organelles such as ribosome, lysosomes, mitochondria, and then give the function or how they work in the cell. So the first one is the cell membrane. The cell membrane is a semi-permeable membrane surrounding the cytoplasm of a cell. It separates the interior of a cell from the outside environment. The cell membrane is selectively permeable to ion and organic molecule and controls the movement of substances in and out of the cells. The basic function of the cell membrane is to protect the cell from its surroundings. It consists of the phospholipid bilayer and with embedded proteins. So there are proteins embedded in the cell membrane, which allows materials such as glucose that cannot go through the cell because of its size. So these embedded proteins help uh, large large molecule to get into the cell. The cell membrane are involved in cell processes such as cell adhesion, ion conductivity, and cell signaling itself as the attachment surface to a cellular structure, including the cell wall and the intracellular cytoskeleton. So we have our fluid mosaic model. The fluid mosaic model describes the plasma membrane of animal cells. The plasma membrane of that surround these cells have two layers. So they are by layers, which are fats and phosphorus attached to it. So which at body temperature are fluid due to their unsaturated nature. And the structure of the plasma membrane supports the old saying, oil and water don't mix. So it's a fluid that is the plasma membrane is hydrophobic, so it doesn't mix with water. The protein and substances like cholesterol embedded in the bilayer gives a looks of the mosaic because the plasma membrane has the consistency of vegetable oil of at body temperature. The proteins and other substances are able to move across it and be fluid. The cholesterol is stacking the membrane more stable and prevent it from solidifying when the body temperature is low. Carbohydrate chains also on in the plasma membrane in the cell membrane attached to the outer surface of the cell membrane, and each cell of each cell. So these carbohydrates are specific to every person and they determine your blood type. So the cell membrane, the fluid mosaic membrane of the cell membrane contains carbohydrate, cholesterols, and proteins transports, and all this good stuff. So they have facilitated diffusion. So facilitated diffusion is first a process of spontaneous passive transport of molecules or ion across a cell membrane through specific transmembrane integral protein. So these are the protein that helps uh, big or ionic substances or molecule to get in the cell. Specific being it this doesn't require act facilitated diffusion, doesn't require energy at all because molecules are moving down to the concentration gradient. So facilitated diffusion is different from free diffusion in several ways. First, the transport relies on molecular binding between the cargo compared to free diffusion. So and the membrane embedded channel or carrier or carrier. The second, the rate of facilitated diffusion is saturable with the concentration difference between the two phases. Unlike free diffusion, which is linear, the concentration difference. Third, temperature dependence of facilitated transport is different due to the activated binding events as compared to free diffusion, where the difference on temperature is mild. So polar molecule large ion dissolved in water cannot diffuse freely across the plasma membrane due to the hydrophobic nature of the fatty acid tails of the phospholipid that make up the lipid barrier. So big and charged particle cannot go through easily, so they have to go through transport membrane, transport protein to get inside the cell. The example of glucose are glucose and iron, sodium ion. So active transport. So these molecules that cannot go directly into the cell and needed the transport protein goes through with also energy added to it. So active transport will occur across the semi-permeable membrane against the concentration gradient. So because it's against, it needs ATP to move against the concentration gradient. So moving from an area of lower concentration to the area of higher concentration, it requires an expenditure of energy released from an ATP molecular energy. Embedded with the hydrophobic heads in the outer layer of the membrane are transmembrane protein molecules to detect and move compounds through the membrane. Transport protein interact with passenger molecule and use the ATP supply energy to move them against the gradient. The carrier molecule combine with the transport molecule such as amino acid and iron to pump them against their concentration gradient. Active transport lets cells obtain nutrients that can pass through the membrane by other means like glucose, which is a very big molecule. Also, there are secondary active transport processes that are similar to diffusion but instead use imbalances in electrostatic forces to move molecules across the membrane. So active transport is especially for these molecules that cannot go through the cell in the cell because of a charge or molecule or because of the size. So these enter the cell through transport protein requiring energy to move them from the out extra environment from the inside to in the inside environment. So our next topic is the nucleus. So the nucleus is a membrane-bound structure that contains, so nucleus contain our genetic material, and it is not only a storage compartment for DNA, but it's also the site of essential cellular processes such as such as transcription, translation, and translation and replication. So the nucleus is the center of the nucleus, and this center of the nucleus assembles ribosomal subunit. A nuclear membrane, also known as a nuclear envelope, is the double lipid bilayer membrane which surrounds the genetic material and nucleus in the eukaryotic cells, and it is semi-permeable. The nuclear pore complexes allow the transport of molecules across the nucleus. This transport includes RNA and ribosomal protein moving from the nucleus to the cytoplasm and protein. Chromatin is mass of genetic material composed of DNA and protein that condense to form a chromosome during nucleotic cell division. So we have our so all the nucleus is mainly contain our genetic material, and it is there are chromatin in our genetic in our gene. So the chromatin is a mass of genetic material composed of DNA the protein that condense to from other. So when DNA protein condense, they form chromosome during eukaryotic cell division. Chromatin is located in the nucleus of a cell during prophase of mitosis. Chromatin fibers become coiled into chromosome, with each chromosome having two chromatid joined at the centromere. So, in conclusion, we can say that the nucleus is the center of the cell and directs every action. So, the rough endoplasmic reticulum is the surface of the rough endoplasm uh, that is third or that contains proteins manufacturing ribosomes. So ribosomes are constantly being bound and released from the membrane. A ribosome only binds to the RER, so the rough endoplasmic reticulum, once a specific protein nucleic acid complex forms in the cytosol. This special complex forms when the free ribosome begins translating the mRNA of a protein destined for the secretory pathway. Amino acids are polymerized and code the signal peptide molecular message that is recognized and bound by a signal recognition particle. Translation pause and the ribosome complex bind to the RER, where translation continues with the protein forming into the RER lumen or membrane. The protein in ER lumen by an enzyme is processed by enzyme which remove the signal peptide. Ribosomes at this point may be released back into the cytosol. The rough endoplasmic reticulum have multiple functions, such as manufacture of lysosomal enzyme with a mannose 6-phosphate mark added in the cis Golgi, manufacture of secreted protein constitutively with no tag or secreted in a regulatory manner involving paired basic amino acids in the signal peptide integral in the signal peptide integral protein that stay embedded in the membrane vesicle, exit and bind to new membrane. Rab protein are key in targeting the membrane. SNAP and snare proteins are key in the fusion of in the fusion. So the rhabdomyoplastic reticulum is where the proteins are made and and synthesized. It's also uh, a network where their protein are synthesized. Just so the smooth endoplasmic reticulum. The smooth endoplasmic reticulum has functioned in several metabolic processes. It synthesizes lipid, phospholipid, and steroid cell, which secrete this product, such as those in testes, ovaries, and sebaceous glands, have an abundance of smooth endoplasmic. So smooth endoplasmic reticulum, we can say, are in smooth cell. Yeah, they're everywhere. They're everywhere. They are in all cells. All cells have smooth endoplasmic reticulum. But they say here that cell which secrete this product, such as those in the testes, ovary, and sebaceous, so they are in smooth cell. So smooth cells secrete all this. It also carries out metabolism of carbohydrate, drug detoxification, attachment of receptors on cell membrane, protein, and steroid metabolism. In muscle cell, it regulates it regulates calcium ion concentration, and the smooth endoplasmic reticulum also contains the enzyme glucose 6-phosphate, which converts glucose 6-phosphate to glucose, a step in gluconeogenesis. It is connected to the nuclear envelope and consists of tubules that are located near the cell periphery. These tubes sometimes branch, forming a network that is reticular in appearance. In some cells, there are dilated areas like the sacs of Ralph endoplasmic reticulum. The network is smooth endoplasmic reticulum allows for an increased surface area to be devoted to the action of storage of key enzymes and the product of these enzymes. So the smooth endoplasmic reticulum is basically has a metabolic process that synthesizes lipid, phospholipid, and steroid in smooth cells. The Golgi apparatus. So the Golgi apparatus is a major collection and dispatch station of protein products received from the endoplasmic reticulum. So it's like a network. It goes from the rough to the smooth to the Golgi. Proteins synthesized in the endoplasmic reticulum are packaged into vesicles, which then fuse with the Golgi apparatus. These cargo proteins are modified and destined for secretion via exocytosis or for use in the cells. In this respect, the Golgi can be thought of as similar to a post office. So it is packages and label item which is then sent to different parts of the cells to the extracellular space. The Golgi apparatus is also involved in lipid transport and lysosome formation. The structure and the function of the Golgi apparatus are linked. Individual stacks have different assortment of enzymes allowing for progressive processing of cargo protein as they travel from the cis to the trans Golgi phase. Enzymatic reaction within the Golgi stacks occur near its membrane surfaces where enzymes are attached. The Golgi apparatus adds amino 6-phosphate labeled to protein destined for lysosome. Another important function of the Golgi apparatus is the formation of proteoglycan. Enzyme is Golgi attached protein to glycoaminoglycan, thus creating proteoglycan. Glycoaminoglycan are long unbranched polysaccharide molecules present in the extracellular matrix of animal cells. So, in conclusion, Golgi apparatus is more like a post office that receives the substances or molecule from the smooth endoplasmic reticulum and then package it and target to send it outside the cell or inside the cell to its destination. Vesicles. So vesicles are made when part of the membrane pinches off the endoplasmic reticulum or the Golgi complex. Others are made when an object outside of the cell is surrounded by the cell membrane. The assembly of vesicles requires numerous coats to surround and bind to the protein being transported. These bind to the coat vesicle. They also trap various transmembrane receptors proteins called cargo receptors, which in turn trap the cargo molecule. Vesicles are small membrane-enclosed transport units that can transfer molecules between different compartments. Most vesicles transfer the membrane assembled in the and the reticulum to the plasma membrane. So how secretory and membrane protein are made using the endoplasmic reticulum. So proteins are destined for secretions are made on ribosome bound to the RAF-ER. The protein move through the endomembrane system and are dispatched from the transphase of the Golgi apparatus and transport vesicles that move through the cytoplasm and then use with the plasma membrane, releasing the protein to the outside of the cell. Examples of secretory proteins are collagen, insulin, and digestive enzyme of the stomach and intestines. Membranes and secretory proteins are synthesized in the rough on the opacity reticulum. There, they are modified. They are modified proteins, protein modifiers. So these protein modifiers modify the protein. Integral membranes uh, protein in the ER is modified by attachment of of a carbohydrate. So vesicle with integral protein part from the ER and fuse with the cyst face of the Golgi apparatus, and the protein passes along the Golgi cisternae. It is further modified by by the addition of more carbohydrates after its synthesis is complete. Its exits as integral it exits as integral membrane protein of the vesicle that birth from the Golgi's transphase, and when the vesicle fuses with the cell membrane, the protein becomes integral portion of that cell membrane. So that's the process. Lysosomes. So lysosomes Lysosomes are cellular organelles that contain acid hydrolase enzymes that break down waste materials and cellular debris. They can be described as the stomach of the cell. So lysosomes digest excess or worn out organelles, food particles that engulf the viruses or bacteria. So the membrane bounds of around a lysosome allows the digestive enzyme to work at the pH they require. So lysosome fuels phagosomes and dispense the enzyme into the autophagic vacuole, digesting the content. They are frequently nicknamed suicide bags or suicide bags by cell biologists, but due to the autolysis. The interior of the lysosome is very acidic compared to the slightly basic cytosol, so it gives a different pH from the cell of the cytosol and to the interior of the lysosome. So a different pH, different environment. The lysosome maintains this pH differential by pumping in protons from the cytosols across the membrane via proton pumps and chloride ion channels. The lysosomal membrane protects the cytosol and therefore the rest of the cell from the degradative enzyme within the lysosomes. The cell is additionally protected from any lysosomal acid hydrolysis that drains into the cytosol, as these enzymes are pH sensitive and do not function well or at all in the alkaline environment of the cytosol. This ensures that cytosolic molecules and organelles are not destroyed in case there is leakage of the isolytic enzyme from the lysosome. So lysosome is a killer. It kills everything that's gone out. It digests. Lysosomal peroxisome. Peroxisomes are organelles involved in the catabolism of a long-chain fatty acids, branch chain fatty acid, D-amino acid, and polyamines. Reductions of reactive oxygen species, especially hydrogen peroxide, and biosynthesis of plasmogens and ether phospholipid critical for the normal function of mammalian brains and lungs. They also contain approximately 10% of the total activity of two enzymes in the pentose phosphate pathway, which is important for energy metabolism. Other known peroxisomal function includes glycolysis in panosomes, glycosomes. So peroxisome can be derived from the endoplasmic reticulum and replicate by fusion. So peroxisome are organelles that involve in the catabolism of very long chain fatty acid, and they're there. They break down oxygen peroxide to form oxygen. Centriole. Centriole is a small set of microtubules arranged in a specific way. There are 9 groups of microtubules. When two centrioles are found next to each other, they are usually at a right angle. The centrioles are found in pairs and move toward the poles, or meaning opposite ends of the nucleus, when it is time for cell division. So centriole mainly plays an important really an important role in cell division. So it helps form the mitotic spindle for mitosis in cells. So centrioles mainly focus on cell division, which helps forms a mitotic spindle for cells during mitosis. So centrioles are always in perpendicular shape in in the larger cytokeratins. So cytokeratins help maintain their shape and internal help maintain cell shape and and cell shape and organization and internal organization. It also provides mechanical support that enables cells to carry out um, essential functions like division and movement. Primarily, it gave the cell shape and mechanical resistance to deformation. The cytoskeleton can also actively contract, thereby deforming the cell and the cell environment and allowing cell to migrate. Moreover, it is involved in many cell signaling pathways in the uptake of extracellular material. Endocytosis segregates chromosomes during cellular division, is involved in cytokinesis, provides a scaffold to organize the content of the cell in space, and for intracellular transport. It can also be a template for the construction of cell wall. Also, it can form specialized structure such as flagella and cilia. 3 proteins inside the keratin are actin filament, 3 to 6 nanometers of diameters, intermediate filament 10 to 10 nanometers, and diameters, and microtubules that are 20 to 25 nanometers of diameters. So we have mitochondria. Excuse me. Excuse me. So we have mitochondria. Mitochondria, it function as the powerhouse, excuse me, of the cells. ATP of phosphorylation of ADP through respiration and to regulate cellular metabolism. So mitochondria is produces energy of the cell. So ATP of phosphorylation of ADP through respiration and to regulate cellular metabolism. The central set of reactions involved in ATP production are collectively known as the citric acid cycle or the Krebs cycle. The intermembrane is the space between the outer membrane and the inner membrane. Because the outer membrane is freely permeable to small molecules, the concentration of small molecules such as iron and sugar in the inner membrane is the same as the cytosol. However, large protein must have a specific signaling sequence to be transported across the outer membrane. So the protein's composition of this space is different from the protein composition of the cytosol. The inner mitochondrial membrane is compartmentalized into numerous cristals, which expand the surface area of the inner mitochondrial membrane, enhancing its ability to produce ATP. The matrix is the space enclosed by inner membrane. It contains about 2/3 of the total protein in the mitochondria. Matrix is important in the production of ATP, with the head of the ATP synthase contained in the inner membrane. The matrix contains a highly concentrated mixture of hundreds of enzymes, special mitochondrial ribosome tRNA, and several copies of the mitochondrial DNA genome. Of the enzymes, the major functions include oxidation of pyruvate and fatty acid and the citric acid cycle. The endosymbiosis theory is that his mitochondria is used to be independent. So the endosymbiotic theory is mitochondria used to be an independent organism that was engulfed by your cells, and how the symbiotic parasitic 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 relationship with the cell. Mitochondria have their own separate DNA and divide on their own. So mitochondria is the powerhouse of the cell. So it provide energy in there. That's all.